Hi everyone, how are you today? I know you must be very busy for this week, and thank you for coming even out of this chaos during your exam week. So, uh, like you can see, during the exam week, I'm not going to collect any scrap paper for attendance checking, so feel free to skip the class. What am I talking about? Because if you are a little worried about this attendance accumulation, the point accumulation, you don't have to worry about during the exam week. So if you are too busy to come over to the classroom during exam week, you can take advantage of this lecture recording instead, I think. Of course, I have to really make sure to be extra careful not to screw up this recording for this matter. And also, as you noticed, hopefully I have uploaded the previous exam questions. Those are the most recent relevant ones. And last year I didn't have any class, and the year before I had some take-home exam for some reason, so that doesn't count. So those. And the final exam question of the one particular year was also uploaded because that content is actually within this boundary of our prospective, the expected exam coverage content. So even that one, probably you might have to, if you have to take a look at. So if you have any question, although I provided some answers with them, but still, if you have any question, you can ask me in person through this any communication tools, or even during next week. I mean, next lecture, the day after tomorrow during your class, or even after the class, we can go over this. Okay, so. Exam will cover until the one I can cover during the Wednesday's lecture. So if I get to finish in the middle of any chapter 4, that will be it. So that only up to that you will be responsible for. Okay. All right. So now if we take a look at the eukaryotic cells. Like I said, actually, the main purpose of objective of this chapter is to take a look at different individual organelles of eukaryotic cells and their functions. But let's take a big picture first. Asshole, sorry. Big picture of what we are going to take a look at. There will be so many different details, miscellaneous details. Some of them are actually unnecessary details at this level. If you are not a biology major, why do you really have to worry about those? But still, overall, with those little details overall, you can draw a big picture about the whole concept, overall concept of an eukaryotic cell. That's why they are also included. So first of all, as I already have emphasized enough, eukaryotic cells, one big difference is a larger cell. Because of this larger cell size, suddenly those eukaryotic cells faced higher demand for energy requirement. So I ended the class, the last week's class, that without the mitochondria, this evolution of a eukaryotic cell would not have been complete, and that is true. So luckily, those cells who wanted to evolve into something different like a eukaryotic cell fortunately obtained one very critical organelle that could provide this higher energy demand, that is mitochondria. So with all those organelles and nucleus, the one distinct characteristic of all these organelles they are all this membrane bound, internally membrane bound. So the membrane, if you studied on your own the chapter, was it chapter 2, the whole biological molecular, or chapter 3 that was on your own, that this all membrane, cell membranes are made of some particular chemicals. We call them phospholipids. And the phospholipid has a very unique characteristics, chemical characteristics. And so it's so ideal to serve as such a barrier like membrane. So all those cells, every cell starts from this outside, the outermost boundary we call cell membrane. But interestingly, the same membrane having the exactly identical chemical composition and organization structure are also found inside of this big eukaryotic cell. So all those organelles are simply nothing but the internally smaller boundary, membrane-bound small territories, so to speak. So you divide further, huge internal space, you divided them using the membrane material internally, and then you just call them, okay, now, from now on, this is something Golgi. And then you do some very specific function within Golgi. From now on, some boundaries we call endoplasmic reticulum, whatever, okay? And inside of which we are going to do some very specific, particular, special metabolism, just like that. Okay. Yes?
근데 그 어떤 원핵세포 가운데서는 오히려 진핵세포보다 더 큰 것도 있는데
Oh,
그건
yes.
왜 그래요?
That's some kind of really surprise and weirdo, huge cell. But that's anomaly. And actually, it's a surprise how they manage it, such a huge cell size. But all average on usually, what we have found is prokaryotic cells are very, very tiny in comparison with the average eukaryotic cells. That's all I can say. And they are evolved from archaebacteria, like I said, and this. So, nucleus. Did I go over this last week? Nucleus? I feel just a deja vu. I felt like, no, I didn't. Oh, you are not sure? Okay, then I will just go over this again. So, nucleus is also enclosed in the nuclear envelope, and it is a place where our genetic materials are stored. And the function, the importance of the nucleus, inside of the nucleus, is that these genetic materials are well organized. And one thing that we want to take a look at is this. Well, it's not convenient. So most of us view our genetic materials are made of this particular structure called the chromosomes. But actually now, let me introduce the newer terms if you haven't heard this yet. Most of these majors, those who took the science course in high school probably already heard of this, something called the chromatins. Okay. So this structure, chromosome versus chromatins, they are like switching back and forth depending on circumstances. What's the difference between chromosome and chromatin? Then we are going to take a look at today, shortly after this. And just by having the genetic material, and if those genetic materials are not doing anything, there's no point. What is the critical importance of this genetic material? They are the one carrying the genes and gene expression. What do you mean gene expression? So those genes, usually the function of the purpose of the genes are to make proteins. Because proteins are all magicians. Having those proteins, we can do anything. Like we can build some structures inside of the cells, or you can do all those catalytic functions like chemical reactions, thanks to this protein-based enzymes. But instead of making proteins directly from DNA, we have to go through some intermediate step that we call the RNA synthesis. RNA is, so to speak, we can view as a copy of the DNA. If DNA is the original, some instruction manual, for example. Did I mention this before? Looks like. So DNA, in the DNA, genes are the one that the instructions for how to make each protein. Just like a recipe for each different dishes. But they are really original, single copy. It's very, very precious one. So you don't want to mess with them. So whenever you want to deal with this recipe, to actually cook something according to this particular instruction, then you always are advised to take photocopies, numerous photocopies of a particular one. That's probably, we can say, the relationship among DNA and RNA and protein. So RNA is the one exact copy of those instructions, and then they come out of the nucleus and then to the cytoplasm, and then behold, okay, here's the gospels. Here is how to make pizza. Here is the instruction for how to make jjajangmyeon. All those different dishes. And those ribosomes, snowman-like ribosome, luckily knows how to read this instruction. And this is the place. That's why we define this ribosome as a site or location of protein synthesis, because this ribosome can read the instruction written on the RNA, which is a copy of DNA, and then assemble one by one. Proteins are what, basically? If you read chapter 3, or if you listen to chapter 3, proteins are made of what? Individual amino acid. So in order to make protein, you have to gather necessary particular order of 20 different kinds of amino acid. Then you build a particular protein. So that's what is going on. So that's the nucleus is just a starting point where this instruction allow the copying process of, and that's copying of this DNA into the form of RNA is what we call gene expression. So if someone asks you from now on, if someone asks you what exactly is gene expression, then it's a copying process of DNA region that function as a gene. So you just copy certain particular region, area of the DNA which function as a gene into exact copy of RNA, into numerous copies, multiple copies, and then you bring them to the cytoplasm where the protein can be made out of those instructions, is what all this about. So these ribosomes are actually mixture of proteins and some particular RNA. We call the ribosomal RNA. And although this whole made complete ribosomes only function in the cytoplasm outside of the nucleus, but actually they are assemblage from individual components being done within the nucleus. Okay. Also. So, the way this genetic material is organized, one particular keyword, important keyword that probably to some of you, at least to some of you, is new, is a nucleosome. It's a type of mixture, kind of associated form of protein, particular type of protein and the DNA. Here, DNA, let's say all these threads are DNAs. And just like this thread, DNA can exist either in this highly packed, compact form, or it can loosen up to spread out form. So, uncondensed form, so to speak. So this particular highly packed structure of this DNA plus some of the protein we call the chromosome. This is chromosome. But actually, so this chromosome as we like generally refer those genetic material, DNA plus genetic material in the nucleus we usually refer as chromosomes, but to be more accurate, this particular chromosome structure can only be seen in a very short window of the time during during a lifetime of a single cell, only just right before the cell division. Just like the day before you move, you start packing your stuff. At least I do. So packing means, so this is a packed form, packing form of your genetic material, DNA, is a chromosome, highly condensed. But during the most of their life cycle or lifespan, DNA, hey, relax. We really don't have to be this tense. Okay? Just go easy. And this is the structure we call chromatin. So they are same DNA material, genetic material, within which some area actually genes are contained in certain area of the whole entire DNA. But depending on your need or circumstances, you can condense this whole DNA structure into this chromosome form, or on some other times you can just relax these DNAs into uncondensed form. Then we call that such a structure as a chromatin. So that is the difference between chromosome and chromatin. Okay. So how do you do that? I mean, what kind of magic can you play to? It's not an accordion like this. You can just squeeze this thing and then expand. How do you do that? Protein. You just invite some particular protein, whose name happens to be histone. This is the name of this protein that they are so friendly with DNA. Chemically, they can easily associate with the DNA molecule. Because the secret is DNA in nature is highly negatively charged, chemically speaking. If you did your homework of studying chapter 3, probably you would have learned this DNA is highly negative. On the other hand, these histone proteins are highly positively charged. So this plus and minus attract each other. Kind of chemical bond. We call them ionic bond. So that's why histone proteins are so easily associated with DNA so naturally, and they can cover the entire surface of DNA. And there is a particular way of this forming this particular structure, and this is called the nucleosome. It's just like one ball-like. Where is the? So this is one childish version of this nucleosome. at the center, the gray center gray ball-like thing represent histone proteins, and around which DNA in yellow color are wrapped. Actually, two and a half round of the wrapping is usually done. So this is the basic structure of a nucleosome. A better version of this nucleosome structure is this here. This is 1 unit of nucleosome. You have eight histone proteins of 4 different kinds. You don't really have to know such details. There are several histone proteins. They're in a single nucleosome unit is just sufficient. And around which this DNA are wrapped. In this way, you can have a highly organized, compact structure. And also the beauty of this nucleosome organization is you can control this distance between each nucleosome unit. Now they are playing like just exactly like accordion. So you can squeeze this or you can expand this. And now if you think this is only in one-dimensional way of condensing and expanding, you may not be overly impressed. So what? Big deal. However, you have to think that this can be done in 3D ways. Vertically and also the other dimension. If you do so, you can really achieve highly compact structure. Initially, if DNA molecule consists of only this double helical DNA structure, if you just lay the naked, so-called naked DNA double-strand structure of a single human cell, it can reach something. The length could be as long as something like 1 or 2 meters. Probably you can. You have really not, you don't have any clear idea about what that means. But let me just, if this was the size of an individual human cell, it should be actually a little smaller than. Then you take out. This is a whole cell, not even a nucleus. Then you take out this whole naked DNA and then you align from here to then it can reach all the way down to like a swan, I guess. I'm not lying. Something, because it can reach up to something 86 kilometer. Huge. So how do you think that that's such a long and huge, enormous material can be packed within this small backpack? It's amazing. That's you can achieve through this chromosome condensation. So that's the importance of this nucleosome structure. And also, it is dynamic. So degree of this condensation, you can control upon your particular needs under the different condition. So probably I will have to say this first. And I really miss eraser function on this. Shit. You come back and the same thing. So whatever. So what's the importance of this adjusting? You can dynamically adjusting this degree of this condensation. It's because it's highly correlated with the gene activity. Within this highly packed, compact, condensed state, you can hardly see any activity in terms of the gene expression activity. So basically, you produce nothing with this kind of structure. And if you think, oh, I need to stimulate, promote some activity within particular region of my particular chromosome area, then you can expand this. Then the overall, general, the gene expression activity will also be increased. So that is the main mechanism by which the cell control different gene expression activity, which is extremely important for multicellular eukaryotes, because you have to have some different activity overall along with your different area of your bodies. That's how you achieve this differentiated tissues and organs and etc. I'm not sure. Most of you don't look happy by now when I just go over. So does that really too difficult for you to follow? Is that the reason why, or just because you are just too tired of this exam week? I know it is not a really easy concept, but these days this concept become more and more important, and also like that's because of that becoming more and more publicized. This concept of gene expression. Gene expression dictates gene expression. Different gene expression will affect your traits. Okay. It's not only the mutation, whether you have particular mutation or not, is the only way you can differentiate yourself from others in terms of a different trait. By also adjusting different amount of a gene expression in particular gene, you can also display different traits. I always take this sad example of this growth hormone. So if you have higher expression of a growth hormone, probably relatively you will have a bigger body. Whereas if you have a smaller expression level of your growth hormone, then you will have a smaller body. But if you examine these two individuals' growth hormone gene, there is no difference in terms of DNA sequence. Then what's going on? People ask. Then how do you still have this big different trait? Because you have a different amount of this gene expression. It's actually very important distinction. And these days, actually, more and more information come out that majority of this gene expression is actually controlled at this level, like chromosome condensation. Adjusting this chromosome structural condensation, you can adjust different amount of gene expression. Yes, that's why I try to bring up this difficult concept, although. So anyway, so then you can adjust this relative distance. So how do you, bottom line is this, how do you make this chromosome condensation in practically achieved? By adjusting distance, relative distance among different nucleosomes. Okay. You can make each individual nucleosome much closer, okay. Like having very close social distance. You are violating social distance rule. Then you will achieve highly condensed, tight packed chromosome structure within which you don't really see very high gene expression activity. You don't anticipate anything. However, on the other hand, if you make each individual nucleosome more distantly, okay, aligned, placed, then you can achieve this less condensed chromosome. Now you call those structures a chromatin instead of a chromosome. So that's the difference. And then now you can enjoy much higher gene expression level within that area. It's not really that difficult to admit. You can just acknowledge it. Okay, whatever. Okay, good for you. So that's how you do it. So then more question might arise. Then how do you do that actually? How do you adjust this? By chemical modification of histones and DNA, like this. So some of the chemicals like methyl and acetyl, okay. Once again, don't pay too much unnecessary details over here. That's for just those details. Let's leave those details to the majors. But simply, if you wondered, how do you achieve this distancing? Okay. By putting some chemical, then this nucleosome can be packed. And by eliminating this particular chemical and then instead putting another chemical, then you can expand this nucleosome so that you can achieve this much less condensed chromosome, which is now chromatin. Is basically what you do, how you can do chemical modification of what? Histone protein, or in some cases on DNA themselves. Usually you can insert some particular chemicals like methyl or acetyl. Those two major, but these are not the only ones, of course. So yes.
근데 그 염색체 모양에 따라서 예를 들어서 원래 이런 염색체와는 다르게 팔이 두 개만 텔로센트릭이거나 메타 무슨 센트릭은 그 염색체 모양이 조금씩 다른 경우가 있는데 그런 경우에 유전자 발현은 어떻게 달라져요?
Doesn't really. Only thing you worry about this level of different gene expression is telocentric or metacentric. It refers to the relative position of the belly area, centromere, right? However, the thing that really affects the gene expression is this area. The end area we call this telomere. And this central area, we call this a centromere. Why do I have to go through this even in this class? These areas are really notorious for such a extremely low gene expression whatsoever. Okay. You don't get to see any too many genes are there. To begin with, there are only very few number of genes, and their activities are usually very low because of this particular structure of chromosome condensation. Did you understand? Or you do? But did you understand? No? Okay, so forget about it. Okay, so those are the chromosome structure. Chromosome versus chromatin in the nucleus. That's one thing. Now, what about the barrier? The barrier, outside barrier of the nucleus, which we call the nuclear envelope, which is made of, guess what? The same phospholipid. This phospholipid bilayer is the standard structure, chemical composition and structure for our cell membrane. So exactly the same thing. Okay. The double layer of water expelling the layer of chemicals we happen to call the phospholipid. If you are not familiar, or even you to the point of so annoyed or uncomfortable about all different names like phospholipid, what the H is that? then probably it's a really, really good time after the exams over. It's good time for you to go back to the chapter 3 and then study. And chapter 3 contains all of such basic, important information about this chemistry of this life. Actually, as a major, or anybody who are seriously interested in this life science, without understanding basic concept of this chapter 3, you cannot go any further. That's all about. Although that's too boring content, that's why I intentionally avoid showing you in the classroom, but that doesn't mean that that's not important. Sooner or later, you will have to face them. So anyway, this phospholipid bilayer, which is exactly the same material as the cell membrane, is the one that make this nuclear envelope too. Okay. And what's the function of this envelope? Whenever this membrane, then always membranes serve as some kind of barrier. Okay. And then control some traffic. Hey, where are you going? Do you have any license to go in? And if someone is trying to go out, some material, hey, wait a minute, where are you going? It's kind of a traffic control is always happening on the membrane. Same thing on the cell membrane, and the same thing on the nuclear envelope membrane too, right? However, there is a big difference between cell membrane and nuclear envelope is this. In the case of let's say this this is a cell membrane, and some material is trying to go in. This is a cell membrane, phospholipid bilayer. This cell membrane is, chemically speaking, is water hating. This is the waterproof barrier. So any material all those material you can divide into two different classes. So one is water friendly, we call hydrophilic. The other ones are water hating, hydrophobic. Yeah. So many materials are hydrophilic, and also so many other materials, chemicals, are hydrophobic. So in this water hating hydrophobic cell membrane barrier, only this hydrophobic material can easily pass through. That includes some kind of a gas. Oxygen, nitrogen, hydrogen, all gases are freely can pass through. And lipids, all those small lipids like a cholesterol, okay, they can easily go in and out. But the vast majority of biologically important materials are all hydrophilic, like glucose, the notorious one, ATP, and all those ions, potassium, sodium, calcium, all those ions. The common characteristic of all these hydrophilic materials are what? All of them are electrically polar, charged, plus or minus. The majority of materials are polar, and we need them. Many of them are nutrient. Many of them are important macromolecules for us to build. So in other words, they cannot go in. And those materials inside cannot be exported across the cell membrane. That's just a big dilemma. How do you manage this? So this is a big problem or kind of a blessing. That's why we need some kind of barrier. Hey, where are you going? I gotta check whether you are safe or not. The cell membrane's function. But at the same time, all these goodies, hydrophilic but important materials, we also have to allow them to pass in. How? So that's why on the surface of the cell there are so many different proteins functioning, specializing in transport. So those proteins, membrane proteins, are there. Those membrane proteins, each different type of membrane proteins are highly specific to each different type of chemicals. So let's say, hey, I am specializing in allowing transport of glucose. Okay. There is a such thing, glucose transporter. And hey, I'm a sucrose. We are both sugars, so can I go in? No way. Like this kind of discrimination. Highly specific. So all different kinds of protein, transport proteins are on the membrane, cell membrane. That's like, by one word, if I were to describe this, then we can say that this is highly selective. Or discriminatory. Am I right? Yeah. So discriminating that my cargo molecule from some other average Jando molecules is the characteristic which is useful, highly important. But here, nuclear envelope, although it also functions as a barrier, but it functions as a non-discriminatory matter. How? They don't employ this particular selective protein transporter. Instead, they simply employ some pores. So anybody's welcome as long as you can fit in this small size, is the way they operate this barrier function. So it's much less stringent, I should say. Okay. So in this way, a lot of material can go in to the nucleus, because for nucleus, like inside of nucleus, there are lots of important activity is going on always, like gene expression. Then you need a lot of raw material to synthesize DNA, synthesize RNA, and once they are done, you have to export those products. So how are you going to do that? Through these nuclear pores. Okay. So this is cartoonic version of one such nuclear pore structure. So there are lots of pores on the surface of nucleus. Within each of small hole pore, materials can come in and go out. Another very important function of this nuclear envelope is this. This barrier does not simply passively function as the blocking material, but also it is like stabilizing axis function as. So to understand such, we need to first understand this concept of a chromosome territory. What is this? Today's lecture is very difficult, isn't it? A lot of new concepts are thrown at you just 1 week before the exam. How cruel is this? Yeah, that is my true personality. Look, this is a human's nucleus, human cell nucleus. And why do you get to see so many different beautiful fluorescent colors? Each different color represents different chromosome. Because you can stain in fluorescent dye, you can stain different type of chromosome with different color dyes, which means even in this stage, so now this cell, the whole chromosome exists in the form of a chromatin, untangled chromatin, not chromosome, individual chromosome, the famous structure of a chromosome. No, this famous structure of a chromosome you only get to see just right before the cell division. So usually you don't get to see this structure. But even that, how do you see this whole distinct, separated so to speak, chromatin or chromosome? You can say. How? That's what we call as a chromosome territory. Oh, we realized recently that, okay, although these chromosomes may seem to exist, like they are all tangled up during the stage of this chromatin, no, that is not actually there. They do have their certain strict boundary that we distinguish as a chromosome territory. What's the big deal? Is that really something important that we need to remember? Maybe. So, this nuclear envelope help to establish each different chromatin establish their own territory by having some particular protein called the nucleolamine protein. Again, you don't really have to pay attention about this name of this particular protein. There are some like particular protein playing like a hole or a pole, not a hole, playing like a pole function underneath of this. If this nuclear envelope is something like the roof-like structure, then there might be some of the structural pole-like structural protein underneath of this, and different chromatin can hang onto this different pole, and that's how each different chromatin can establish their own small local territory. Is probably you can basically understand. But understanding their different structures is not important. But why maintaining this chromatin structure is so important, significant biologically, is the issue. So this is some description, pictorial description of each different chromosome territories. Yeah. So this is something, the real thing. And if you mess up with this, then you, unfortunately, you can suffer from this devastating disease, illness. So recent newspaper, I read that this progeria, this is a progeria, the genetic disease, which showing the symptom of premature aging. So all these progeria patients cannot pass their lifespan, cannot pass their teens usually. So when they usually about to reach their about 20s, and usually they pass away because of their physical age is something like those of 80s and 90s years old people. And so why such a horrible symptoms can be found by this progeria patient? What caused this progeria is really silly cause, so to speak. It's a defective protein, one of these proteins that function as a pole underneath of this nuclear envelope. So genetically you have a defect, so you don't get to produce this particular protein, so you don't have any this pore, so nuclear envelope will collapse. The structurally unstable nuclear envelope collapse. So you can see this is, so to speak, normal. I really hate using the term normal, but let's just put this normal nuclear envelope. And this is a progeria patient nuclear envelope, which has a crooked, collapsed structure. And what happened is, once you have this collapsed structure, then this is one particular chromosome, the position is shifted into different positions. So chromosome position can move into different location because of that. And that's all. You don't have anything, any other defect on this progeria. And that is such a big deal of it. Why? Because it will affect. If you move a certain chromosomes area, location into different part of the nucleus, then totally you screw up the highly finely programmed order of gene expression. Once again, gene expression. So this gene expression patterns are disrupted, and that is the reason why you develop such a horrible defect, disease. So not only the defect and the serious deleterious outcome due to the position change of a chromosome, not only found only in the progeria, but also, for example, in cancer. Typical symptom of this cancer is particular chromosome position changed into different area positions. And also the famous, notorious immune disease also can you find also this chromosome position shift. So once again, overall, this relative location or position of a particular chromosome turns out to be extremely important one. So you don't want to mess with such a thing. You just really preserve. Try to be so careful about this. Otherwise, you will disrupt all your gene expression order, and you will face the consequences. Is the take-home message out of this. So that try to answer this question: why is a collapsed nuclear envelope so deleterious? Because of this disorder of gene expression. If you disrupt the chromosome territories, then you will result in this disrupted gene expression order. Is this? Okay, that's so much about nucleus. Now let's switch the gear into plasma membrane. Plasma membrane is one of the most famous the external barrier for us. Okay. So that is the starting point of any living life. Cell, if you want to make cell, and you have to draw the line first. Hey, from now on, this is a territory, inside of which is my own territory. No one. I don't want to see anyone to mess with this, my holy area is actually the concept. And the next thing is whether you can have this or not. If you are lucky enough to capture this genetic material, then you are lucky, so that you may be able to live forever with this genetic material. That's the starting point of all this generation of spontaneous generation of life in the form of a cell. But anyway, so this cell membrane function as a barrier. However, this is not merely a simple barrier, but it is doing much more than this. Due to channels and transporters are all those proteins. Channel, whether you call this as a channel or simply you call them as a transporters, they are all proteins on the membrane that function as a transporting some particular material. Hey, what's this channel? Why do you call some different transporting channel, protein as a channel? I don't know, but channels usually means the ions like sodium, potassium, all these charged ions. When a certain protein function as a transporter for these ions, then you, by convention, you call them as a channels specifically. So from now on, outside you get to hear, oh, some sodium channel, potassium channel, calcium channel. Those are, oh, I see. They are the transporter proteins for these specific ions. Then probably you can understand this. And so this plasma membrane, universal means from bacteria to human, if you examine the plasma membrane chemically, they are all, you can say they are all identical. In terms of, what do you mean by identical? So they are all made of so-called phospholipid. Phospholipid, two layers of phospholipid, we call it a phospholipid bilayers. So bacterial cell membrane and human cell membrane, everybody's cell membranes are basically made of the same chemical composition we call as a phospholipid bilayer. But at the same time, distinct means even in the human body, if you look at different area of tissues, then the fine details of this chemical composition of this phospholipid bilayers are different. So those are the areas that you don't worry about, but simply, okay, so overall they are the same, but in details, not exactly each different cell membranes are 100% identical is probably what you can, how you can understand this phrase. And unique molecular markers, this concept is also, although it is not easy, it may not be easy, but very important in that each different cell has to carry their different name tag. Otherwise, how can you distinguish some foreign invaders, okay, from one of your own? Let's say you are patrolling white blood cell, and your job is to find out some intruders, then select them out and destroy them. But unfortunately, as a white blood cell, you don't have any eyes. Okay? How can you identify anybody who is foreign? Like the famous example of a different blood type. Type A blood, type B blood, type O blood type, type AB blood types. I mean, based upon what do you distinguish this different blood type? Is it based upon some different markers present on your surface of red blood cells? So cell surface, cell membrane carry very unique different recognition markers in the form of, tell you what, in the form of carbohydrates. So different cell in my body, in your body, different type of cell carry different shapes, different characteristic of carbohydrates, which are very unique to each different type of cell. They carry them so that they can display, they can present themselves, hey, I'm a red blood cell. Don't you recognize me? Like, hey, I'm a fat cell. Hey, I'm a brain cell. Type of, every different cell carry these specific markers. Carbohydrates is extremely fitted for such function. Carbohydrates are. Protein cannot do this with flexibility like carbohydrates. So carbohydrate, if you thought that carbohydrates are just for like eating, they are serving you lunch or dinners. A lot of people actually avoid carbohydrate for at least for your dinner because of this weight control problem. But anyway, so no. Then think again. Carbohydrates are very valuable as an energy provider. Also, this type of specific recognition markers. So that's present on the cell surface. Probably, if you read this thing, basically, just emphasizing once again one particular point of the previous slide. That's all. Okay. What's this fluid mosaic model? This is one cartoon, representative cartoon of a portion of cell membrane. So phospholipid bilayer means this is one such phospholipid layer on top. The bottom, identical phospholipid bilayer which flipped, actually. If you look carefully, these chemical structures are like facing opposite side of. So these are two layers, but top layer and bottom layers are like facing the other orientation. But they are double layer, bilayer too. And there is a reason why they have this particular configurations. Go back to your chapter 3, that is well explained. So anyway, so here, this phospholipid, basically this phospholipid bilayer behave like a semi-liquid, like a fluid. As a lipid, whole bunch of lipid that kind of behave. You can view this thing as a liquid dish. And this proteins embedded in the membrane, just like embedded the almond, like so milk chocolate. It's not only pure milk chocolate, but almond milk chocolate is a cell membrane that is. And these embedded proteins so usually can float, not in high speed, but slowly they can float along this liquid-like cell membrane. That's what this so-called fluid mosaic model describes. Although it is really a famous phrase coined some time ago by probably some famous cell biologist. But practically speaking, in these days, who cares about whether this is a fluid mosaic model or what not? But anyway, that's one of the such a famous phrase. So probably if someone asks you, hey, what's fluid mosaic model? You took a life science course or anything, and probably you can explain this much. Oh, that's nothing. And another thing you can, what else? So let's examine this as one of the templates of a typical cell membrane. Although this particular model is about eukaryotic cell membrane, however, the prokaryotic cell membrane is not much different from this. Basically, very similar. Anything else you discovered here? So you have so many embedded proteins. Okay. What would you say that if I asked you what's the major function of all these embedded proteins? If I ask you, hey, what's the general function of these all embedded proteins on the cell membrane? What do you say? I just shouted for an hour long, then you cannot even say one major function of this embedded protein on the cell membrane. Pity. Okay, great. Thank you. Transporters. There are so many proteins probably acting as a different transporters. for different type of chemical molecules on the cell membrane. That's why you need so many different types of proteins. You need them, right? But that's not all, of course. Some other proteins are not acting as a transporters, but also probably act as a receptors. Once I showed earlier that the difference between receptor and transporters. Receptors are only recognize the signal, does not pass this molecule through this, but simply recognize, oh, okay. Like, for example, the difference between glucose transporter and glucose receptor. What do you say? Glucose receptor on the cell membrane can only detect the presence of glucose outside, okay, and then capture, oh, there are glucose. And then they can sense the abundance, availability of glucose, and then send the message to the cell. Hey, I detect so many glucose. That means we have plenty of food available these days. So we can be relaxed, okay? We can be a little bit more luxurious, if you will. Type of signal you can send them. Glucose transporters, on the other hand, directly actually transfer this glucose molecule from one area to the other area. Actually, import. You can import. So that's the difference between transporter and the receptors. And also, the antibody protein. Antibody proteins are also present on the cell membrane too. Some of them. And so many different types of molecules, and even some of them act as the enzymes in the cell membrane too. So you name it, you can imagine so many different types of proteins are present in the cell membrane. So having said that, on this, at least on this particular cartoon, do you see any one peculiar tendency, one particular observation you can make on this cartoon? Anybody? What are these? The green thingy. The little green thing. The little green antenna-like things. What do you say? These are, guess what? These are, you have no idea? Green, cute. They are carbohydrates that I mentioned earlier today. A marker, recognition marker, signal. They are carbohydrate. So those, what's this glycoproteins? The name of glycoprotein, anyone? What do you mean by glycoprotein? Protein that having this carbohydrate attached. Glyco means carbohydrate. Protein means, so this is a protein, but having carbohydrate attached. So there are so many glycoproteins. But one thing is, how come you only get to see this carbohydrate only facing outside? So this is outside and this is internal, inside the cytoplasm side. Always these carbohydrates on the cell membrane, okay? The carbohydrate on the cell membrane protein are always facing outside. Why? That is something to do with the carbohydrate function. What did I say about the function of a carbohydrate over here? Recognition, name tag. So when somebody else, like a white blood cell, try to identify you as a cell, whether you are one of us or some foreign invader, would this white blood cell try to stick his hand inside of this cell membrane and try to scan, you are one of us? Or would this white blood cell try to scan the surface like, oh, okay, you are one of mine? That is the case. That's why all these carbohydrates should face outside on the cell membrane. Okay? And do you see another differences in this cartoon? So we can actually analyze so many different things by simply digesting this little simple cartoon. Another different observation you can point out would be, it seems like there are two different types of proteins on this figure. One type of protein is called integral membrane protein. The other one is called peripheral membrane proteins. The differences are physical appearance-wise. The difference is the integral membrane proteins are always stuck in the middle. They're embedded in the lipid bilayer of the cell membrane, whereas peripheral membrane proteins are, although they can be found also in the cell membrane, but they never stick themselves physically in the middle of this cell membrane, but simply they're associated, attached to the surface of this membrane. Those are called the peripheral, and those who actually insert its body itself in the middle, into the middle of this cell membrane is called integral membrane protein. Why do I make this distinction? It's because it makes really big significant difference in terms of their synthesis and in terms of their transport. Okay, so to give you a little preview today only, integral membrane proteins are, the reason why they can stick themselves in the middle is because it contains this. Something that, so chemically speaking, this middle layer is what? How do you define this middle layer of this cell membrane? Whether it is hydrophobic or versus hydrophilic? What do you think? Is it going to be hydrophobic or hydrophilic? Hydrophobic. Cell membranes are hydrophobic. And the reason why they are hydrophobic is this thick middle layer. They are lipid layer. Okay. So they are hydrophobic. Any hydrophilic material cannot even get closer to this. Go away. Who are you? Hydrophilic? How dare you? Can you approach me? So only hydrophobic molecule can be associated, can be inserted into this. Proteins, most of the proteins, once again, the chapter 3 explains, but most of the proteins on the surface are hydrophilic proteins. Why? Because their surrounding environments are all what? Water. They are all water. Hydrophobic, hydrophilic means it's just a relative term related to the water. Hydrophobic means water hating because they are lipid, non-polar. Hydrophilic means they are polar, charged. So they can establish friendly association with water molecule because water molecule is also polar. So most of these proteins are surrounded, the surface is at least surrounded with the water molecule. So when proteins are initially made, proteins are unique in that different area of a protein have a different chemical properties. Like, let's say this is a hydrophobic area, your belly, and this, let's say this is a hydrophilic area of a protein. Then once the protein is made, just finished the protein synthesis, and they spontaneously fold itself to hide this hydrophobic area, so that they can exclude the contact, the water, contact from water, this area. And only those area who can handle the the water can remain on the surface. That's how protein shape is determined, basically. So anyway, basically all these proteins are hydrophilic, but very special proteins can still have this hydrophobic area retained and exposed outside. And those type of protein can be inserted in the middle of the cell membrane. And this particular structure we called as a transmembrane alpha helix. This alpha helix is one particular shape of a protein. Okay? And this is something, this transmembrane alpha helix, not all alpha helix, but this particular transmembrane alpha helix is hydrophobic. So that's why using that, this integral membrane protein can behave as an integral. Okay, I have to stop and just from the atmosphere I have a feedback. I don't think this is a kind of easy for you to take. So, how am I going to do with this? But anyway, we have one more lecture to go about this until the midterm exam. So I will stop today and then see you on Wednesday.